Hello there, welcome! Today we've got two exam questions on protein electrophoresis and they're in the application style. They're a bit hard as well, so try your best, pause the video here, try these questions out and continue on to see how I answer them. Okay, I hope that you guys are very well versed in proteins, what charges they have, how they differ, because it will help you a lot with these questions. So the first one says, suggest why proteins are heated before being placed in an electrophoresis gel. Number one, you zoom in, okay? Zoom into what the question says. Why are proteins heated? Well, without knowing what the context is, we can immediately say denaturing, right? You want to denature the proteins. But then it says why, right? Let's tap into that a bit more. Well, you see, proteins have negative and positive charges, and so by denaturing it, it will expose the charges on the proteins, which is really good. Why? Because in electrophoresis, that's how, it can move up the gel. It works in the whole electrophoresis process, and that's why we need it to be denatured in the first place, to expose the charges. And then second question says, suggest why the binding of SDS to proteins is necessary for protein electrophoresis. So I've written 4 points there, we only need 2. Essentially with this one, it's a suggest, right? So suggest questions is based on content, but also you kind of need to think a bit more about it. But with suggest questions, I normally like to recommend reading the information. So let's read it. Mixtures of proteins can be separated by electrophoresis. Proteins are heated before being placed in the electrophoresis gel. The gel contains a substance called SDS, which has a negative charge. Let me repeat that again. A negative charge. And then SDS binds to proteins. Let's rephrase the question. Why is it that SDS binding to proteins must happen? Right. You might not be able to answer that immediately, but you can once you've identified and realised it said this part, negative charge. Remember when I told you earlier proteins have different charges, right? And that's going to interact with the whole buffer, the solution, and it might actually hinder the protein from moving up the gel. And so because SDS magically has been added, it allows for all the proteins to become negatively charged, so they're all uniform, right? All the charges are the same because they are negative, and then that means that the proteins can move in the same direction, as I've written here. There's other points that different proteins have different overall charges. This is what I was on about. That's a mark, Another point is proteins will be separated by mass. I wouldn't have said that as an answer, but hey, it's in the mark scheme. So yeah, the ones I've starred are your, say the sentences that really answer the question. So if you want to use this part I as a sort of question for your notes for understanding, then those 2 starred sentences are perfect for that. So yeah, when you you see application questions which come very frequently, because this is hello A-levels, you really want to tap into what you know about the process and then how does it link with what they're saying, because there is a link. There's a reason why the examiners made these questions in the first place. They're testing your knowledge in a different way, and when they've given you something that's unknown, aka SDS, look at exactly what they say about it, because that will probably actually 100% of the time be the answer that they're looking for. For example, in this one, the fact that it's negatively charged, look how much we could extract from that and be able to answer it. So yeah, remember the 2 most important exam technique tips are read the question first and state all the obvious points. Now over to you. Try some questions and let me know how it goes.